The human brain is an organ of extraordinary complexity, weighing about 3 pounds and serving as the command center for every function, sensation, and thought that defines our lives. It interprets the world around us, orchestrates our movements, and shapes the very core of our behavior and memories. Nestled securely within the cranium, the brain is not only the seat of intelligence but also the interpreter of the senses, the silent conductor that initiates movement and governs behavior. Its architecture, though intricate, can be understood by examining its fundamental regions and the structures that support and protect it. At the broadest level, the brain is organized into 3 primary regions: the cerebrum, the cerebellum, and the brainstem. Each plays a distinctive role, yet all operate together, forming a seamless network that controls every function of the body and embodies the essence of the mind. The cerebrum, the largest and most highly developed part of the human brain, dominates the cranial cavity. Within the folds and grooves of the cerebral cortex, higher functions such as sensory perception, information processing, and voluntary movement originate. The cerebrum is divided into 2 hemispheres, right and left, each responsible for processing information from the opposite side of the body. These hemispheres are united but also possess specialized functions, contributing to the versatility and adaptability of human cognition. Beneath the cerebrum lies the cerebellum, smaller in size yet vital in its responsibilities. The cerebellum, situated under the cerebrum, is primarily tasked with coordinating muscle movements, maintaining posture, and preserving balance. It is deeply involved in learned movements, ensuring that our actions are smooth and precise. The cerebellum contains the highest concentration of neuronal cells in the brain, supporting its essential role in refining motor activity. Connecting the cerebrum and cerebellum to the spinal cord is the brainstem, a structure that serves as a crucial relay center. The brainstem is composed of the midbrain, pons, and medulla. It acts as a bridge between the higher brain regions and the spinal cord, facilitating the passage of information to and from the brain. The brainstem is responsible for many of the body's automatic functions, such as breathing, heart rate, and the regulation of vital processes essential for survival. In essence, while the cerebrum allows us to think, feel, and plan, the brainstem ensures that the fundamental rhythms of life continue without conscious effort. Delving deeper into the composition of these regions, we encountered 2 key types of brain tissue: gray matter and white matter. The brain's outermost layer, known as the cortex, is composed mainly of gray matter. Gray matter houses densely packed neurons and forms the substrate where the brain's electrical activity unfolds. Here, in these rich networks, neurons fire, blood flow shifts, and metabolic activity surges whenever the brain is engaged. The cerebral cortex, with its characteristic gyri and sulci, those ridges and grooves that give the brain its folded appearance, maximizes the surface area available for gray matter, accommodating a vast number of neurons within the confines of the skull. This expansion of surface area is a hallmark of human brain evolution, supporting our advanced cognitive abilities. Beyond the cortex, gray matter is also found in deeper brain regions, where clusters of neurons known as nuclei play specialized roles in processing and relaying information. The cerebellum, too, boasts an abundance of gray matter, contributing to its ability to fine-tune movement and balance. White matter, by contrast, resides primarily in the deeper layers of the brain. It is composed of myelin-covered axons, the long, slender projections of neurons that serve as communication highways between different parts of the brain and between the brain and spinal cord. White matter tracks interconnect regions of gray matter, allowing for rapid and efficient transmission of information. The integrity of these white matter pathways is crucial. Evidence suggests that damage to highly connected regions of white matter may be more predictive of cognitive impairment than damage to gray matter hubs. Thus, white matter is not merely structural. It is the infrastructure that enables the brain's many parts to function as an integrated whole. The brain does not exist in isolation. Its delicate tissues are enveloped by a series of protective structures that shield it from injury and infection. Closest to the brain itself are the meninges, a trio of membranous layers that surround both the brain and the spinal cord. These 3 layers, the dura mater, arachnoid mater, and pia mater, serve as both armor and support. The dura mater, the outermost layer, is thick, tough, and composed of dense connective tissue. It adheres to the inner surface of the skull, providing a formidable barrier against trauma. The reflections of the dura mater help divide the cranial cavity into compartments, offering further protection to the brain's delicate regions. Beneath the dura mater lies the arachnoid mater, a web-like membrane that acts as a cushioning layer between the dura and the innermost covering. The innermost layer, the pia mater, is thin and delicate, adhering closely to the contours of the brain itself. These meninges not only protect the brain, but also provide a support system for blood vessels, nerves, lymphatics, and the flow of cerebrospinal fluid. They act as shock absorbers, buffering the brain against the minor impacts and pressures that might otherwise cause injury. Encasing all of these layers is the cranium, the bony structure that forms the skull. The cranium's rigid walls are the first line of defense against external forces, helping to prevent injury to the brain's soft tissues. Between the skull and the brain, the meninges and cerebrospinal fluid form additional layers of protection, ensuring that the brain remains as safe as possible from the hazards of daily life. The architecture of the human brain is further reflected in the way it connects with the rest of the body. The central nervous system, which comprises the brain and spinal cord, is distinct from the peripheral nervous system, which includes the spinal nerves and cranial nerves that branch out to every part of the body. Through this extensive network, the brain communicates with distant organs, muscles, and sensory systems, orchestrating the complex interplay that underpins every sensation and movement. Despite centuries of study, certain details about the deep organization and interaction of these regions remain topics of ongoing research. Scientists continue to investigate the precise ways in which the cerebrum, cerebellum, and brainstem communicate, and how the integrity of gray and white matter supports the emergence of consciousness and behavior. While the broad outlines of brain anatomy are well established, the intricate choreography of its internal workings is still being unraveled. In understanding the foundations of brain structure, we see not only a marvel of biological engineering, but also the profound interconnectedness of protection, communication, and control. The interplay between the cerebrum, cerebellum, and brainstem, the essential roles of gray and white matter, and the vigilant guardianship of the meninges and cranium all serve to support the organ that makes us who we are. This architecture, at once robust and delicate, is the stage upon which the drama of human life is played, a testament to nature's ingenuity and the enduring mystery of the mind. Imagine for a moment that you could shrink yourself down and wander across the surface of your own brain. What you would encounter would not be a smooth landscape, but a dramatic terrain of deep valleys and towering ridges, a wrinkled sheet of living tissue covering the brain's outermost layer. This is the cerebral cortex, an intricate, convoluted expanse known as gray matter, just a few millimeters thick yet packed with billions of neurons. The folds and creases, called gyri and sulci, are not mere quirks of nature but ingenious adaptations. They allow the cortex to fit a vast surface area within the limited space of the skull, maximizing the brain's ability to process information without requiring a head the size of a pumpkin. But the brilliance of the cortex goes beyond its structure. This thin, rippled sheet is divided into distinct regions, traditionally described as four lobes: the frontal, parietal, temporal, and occipital. Each lobe is like a specialized neighborhood, bustling with unique functions and responsibilities, yet all are interconnected by dense networks of fibers. The boundaries between these lobes are marked not by fences or walls, but by major grooves in the cortex. While each lobe tends to lead in certain tasks, their collaboration is constant and essential. Let us begin at the front, with the frontal lobe. Positioned just behind your forehead, this region serves as the command center for what scientists call executive functions. Here, the brain orchestrates planning, reasoning, and problem-solving abilities that let us weigh consequences, set goals, and imagine scenarios before acting. The frontal lobe also directs voluntary movement with astonishing precision. It is here that our sense of self, personality, and ability to navigate the social world take shape. The story of Phineas Gage, though not explored in detail here, has long shown how injury to this area can profoundly alter a person's behavior and temperament, a testament to the frontal lobe's vital role in shaping who we are. Shift your attention a bit further back and you enter the parietal lobe. This region is a master integrator, where the senses converge. The parietal lobe receives and assembles information from touch, temperature, and spatial awareness, constructing a map of the body's position in space and its relationship to the world. Thanks to this lobe, we can reach out to grasp an object, feel its texture, and judge its weight, all without consciously thinking about each step. The parietal lobe's ability to blend and interpret sensory data underlies much of what we experience as perception, and its seamless integration with other brain regions is essential for our everyday interactions. On the sides of the brain, just above the ears, lie the temporal lobes. These regions are deeply involved in processing sounds and language, deciphering the intricate patterns of speech and music. But the temporal lobes do more than handle auditory information. They are also crucial for memory. Within their depths, the brain stores and retrieves the events of our lives, drawing on past experiences to inform present decisions. The temporal lobes help us recognize faces and understand the emotional tone of voices, weaving together strands of perception, memory, and meaning. At the back of the brain sits the occipital lobe, the primary center for visual processing. Here, raw signals from the eyes are transformed into the rich, detailed images that fill our conscious experience. The occipital lobe sorts colors, shapes, and movement, building the visual world piece by piece. While it specializes in vision, its work is far from isolated. The occipital lobe constantly communicates with other lobes, ensuring that what we see is integrated with what we hear, touch, and remember. Traditionally, these four lobes are the main characters in the story of the cortex, but the landscape is even more complex. Some neuroscientists recognize additional regions, including the insular and limbic lobes, which are folded deep within the cortex. These areas contribute to functions like emotion, self-awareness, and the regulation of bodily states. The boundaries and exact roles of these additional lobes are ongoing areas of study, but their existence hints at the layered complexity of the brain's outer shell. Looking closer, the cortex is not a uniform sheet, but a layered structure. The most prominent part, known as the neocortex, consists of 6 distinct layers, each with its own types of neurons and patterns of connections. These layers are stacked like sheets of paper, just 2 to 4 millimeters thick in total, but with a surface area that spans hundreds of square centimeters. Within these layers, the business of perception, attention, memory, and consciousness unfolds. Some layers receive incoming sensory information, while others send signals out to the body or to deeper brain regions. The precise arrangement of these layers allows for the flow and integration of information, giving rise to the brain's remarkable versatility. Information does not flow through the cortex in isolation. Buried deeper within the brain are the subcortical regions, a collection of diverse structures that act as hubs, relays, and regulators. These subcortical formations include the thalamus, which serves as a crucial way station, relaying sensory data to the appropriate cortical areas. The basal ganglia, a group of interconnected nuclei, play fundamental roles in motor control, helping to smooth out movements and initiate voluntary actions. Other subcortical structures, such as the hippocampus and amygdala, are central to learning, memory, emotion, and even the experience of pleasure. The influence of subcortical regions on the cortex is not limited to moments of intense action or emotion. Even during periods of rest, these deep structures modulate cortical activity, shaping patterns of connectivity that underlie our ability to think, feel, and interact with others. Research suggests that abnormalities in these subcortical regions are associated with a variety of psychiatric and neurological disorders, highlighting their pivotal role in both health and disease. Subcortical gray matter structures, including the putamen, pallidum, caudate, thalamus, amygdala, and hippocampus, are involved in an array of processes. They regulate not only movement and sensation, but also decision-making, language development, and the full spectrum of cognitive and social functions. In many ways, these regions are the brain's backstage crew, supporting the performance of the cerebral cortex and enabling the higher-order abilities that define the human experience. The division of the cortex into lobes and the layering of its neurons, together with the interplay of cortical and subcortical regions, illustrate a central principle of brain organization. Function is mapped to form. Each lobe and layer brings its own specialization, but none acts alone. The true marvel of the brain lies in the seamless coordination of its parts, the constant exchange of signals across networks that span from the surface of the cortex to the deepest subcortical core. As we map the architecture of the human brain, we glimpse the outlines of a system that is at once structured and flexible, specialized and integrated. The cerebral cortex, with its lobes and layers, provides the scaffolding for thought, sensation, and action, while the subcortical regions lend depth and nuance to our inner lives. Together, they form a living mosaic, one that, despite generations of study, continues to astonish and humble those who seek to understand it. The journey through this landscape has only just begun, and with each new discovery, we come a little closer to unlocking the secrets of how the brain's physical form gives rise to the rich tapestry of the human mind. If you could watch the brain at work, not as a static object but as a living network, you would witness breathtaking dynamism. No single neuron acts alone. The brain's true genius lies in its ability to forge connections, billions of neurons weaving a tapestry of communication, forming vast and intricate networks that span its landscape. These connections are not random. They are carefully sculpted highways and byways, built over a lifetime, guiding the flow of information and shaping who we are. Neurons, the nerve cells at the heart of this system, serve as messengers. Each neuron extends branching fibers, dendrites to receive incoming signals, and a long axon to transmit its own message onward. When a neuron fires, it generates an electrical impulse, an action potential that zips down the axon like a tiny bolt of lightning. But what happens when that electrical surge reaches the end of the line? At the axon terminal, something remarkable occurs. The message shifts from an electrical event to a chemical one. Tiny sacs release neurotransmitters, chemical messengers, into the narrow gap known as the synapse. These molecules drift across and bind to receptors on the neighboring neuron, triggering either an excitatory or inhibitory response. In this way, the signal leaps from cell to cell, carried forward by a relay of chemistry and electricity. Most neurons are loyal to a single type of neurotransmitter, releasing it at all their synapses. The nature of the neurotransmitter, whether it excites the next neuron or holds it back, shapes the flow of information. Glutamate, for instance, stands out as the brain's principal excitatory neurotransmitter, effectively pushing the next neuron toward firing. Others serve to dampen activity, fine-tuning the overall balance of excitation and inhibition essential for healthy brain function. This chemical symphony is not just a way to get messages from point A to point B, it is the foundation of movement, sensation, memory, thought, and emotion. Even the simplest muscle twitch or fleeting feeling emerges from the release and uptake of these molecules. Yet it is the pattern of connections, the architecture of the networks themselves, that transforms a collection of cells into a thinking, feeling brain. During development, the human cortex forms about 100 trillion synapses. These are the points where axons from one neuron meet the dendrites of another, creating a vast web of potential communication. Some connections are local, forming short circuits within a patch of gray matter. Others stretch out as long-range projections that make up the brain's white matter, linking distant regions into unified networks. The arrangement of these pathways is not haphazard, it defines the brain's functional territories. For example, functional areas in the cortex can be mapped by tracing the major association fibers that link them, revealing that a region's role often depends as much on its connections as on its internal machinery. Advances in neuroimaging have allowed scientists to map these networks in exquisite detail, offering a multimodal perspective that combines structure and function. High-resolution scans reveal not only the shape and thickness of gray matter, but also the degree of myelination, the patterns of gene expression unique to each region, and even the distribution of neurotransmitter receptors. This level of detail has opened a window onto the central nervous system's organization, helping to explain why certain areas are specialized for vision or language, while others become hubs for memory or emotion. Within the brain, neural pathways are the roads and highways along which information travels. Some are ascending or descending tracks that bridge the brain and spinal cord, ferrying sensory input upward and motor commands downward. Others, such as those within the limbic system or basal ganglia, form deep intracerebral loops, essential for regulating emotion, motivation, and behavior. At the smallest scale, microcircuits handle rapid automatic tasks like reflexes or the first steps of sensory processing. At a broader scale, macrocircuits connect clusters of neurons across lobes or even hemispheres, enabling higher functions like reasoning, creativity, and self-reflection. The brain's ability to communicate internally is not simply a matter of shuttling signals back and forth. It is a feat of coordination, an ongoing negotiation among countless specialized regions, each contributing its strengths to the collective enterprise. Functional imaging studies have shown that when we perceive, move, think, or feel, many brain areas light up in concert. These patterns of activity can be mapped to specific networks, circuits for attention, others for memory, language, or emotion. The collaboration among these networks underpins our most complex abilities. Emotion, for example, is not housed in a single spot, but organized through large-scale network interactions that span cortical and subcortical systems. The limbic system is a key player, yet it does not work in isolation. Instead, it engages in dialogue with regions of the cortex and other deep structures, regulating feelings, memories, and motivations in a constantly shifting interplay. This interdependence is not unique to emotion. Cognition, perception, and even basic sensory experiences rely on the dynamic integration of multiple regions and pathways. Connectivity is a central concept in modern neuroscience. It refers to the influence that activity in one brain region has on another, and it is this influence more than any single structure that enables the brain to adapt, learn, and recover from setbacks. When researchers map connections at the mesoscale between individual neurons and large-scale networks, they gain insights into both healthy function and the origins of neurological or psychiatric disorders. Sophisticated modeling tools, such as graph neural networks, now allow scientists to analyze these complex webs, integrating data from diverse imaging techniques to better understand both normal and pathological brain activity. The brain's networks are not static. They are shaped by experience and constantly refined through a process known as neuroplasticity. This remarkable property allows the nervous system to alter its structure, function, and connections in response to stimuli. Neuroplasticity operates through several mechanisms: synaptic plasticity, in which the strength of connections changes; structural plasticity, which involves the growth or pruning of connections, and even entire neurons, neurogenesis, the birth of new neurons in select regions like the hippocampus; and functional reorganization, as networks adapt to new demands or compensate after injury. This ability to rewire is not just a feature of childhood. Even in adulthood, repeated thoughts and behaviors can strengthen certain pathways, while unused ones may fade away. Plasticity allows the brain to recover after trauma, adapt to sensory loss, or learn new skills, demonstrating a resilience that is central to our capacity for growth. Another intriguing aspect of brain connectivity is the way it supports shifting modes of attention. The brain is not always focused outward, processing information from the external world. At times, it turns inward, recalling memories, planning for the future, or letting the mind wander. Different neural dynamics support these modes, with networks coupling and decoupling as needed. When the brain attends to internal thoughts, the patterns of connectivity shift, temporarily reducing responsiveness to external stimuli. This flexibility allows us to construct internal models of the world, supporting perception, prediction, and action. The architecture of the human brain is thus best understood not as a static map, but as a living network, one that is both highly structured and endlessly adaptable. Different neural populations, linked by complex circuits, interact to support perception, movement, thought, and emotion. The efficiency with which these networks are organized and communicate may even underlie what we recognize as intelligence. Each moment of sensation, each movement, each fleeting feeling or memory is an emergent property of this collective activity. In mapping the brain's connectivity, neuroscience continues to unravel how structure gives rise to function, how local circuits contribute to global states, and how the sum of these interactions shapes the experience of being human. The story of the human brain is not one of static structures or rigid blueprints. Instead, it is a tale of constant integration, adaptation, and response, a living architecture designed for the unpredictable theater of real life. In laboratories and clinics, controlled experiments have long served as windows into brain function, yet only in recent years have scientists begun to observe the brain in action as people navigate the world, make decisions, laugh, worry, and connect with others. Functional MRI. Studies conducted in naturalistic settings, while people converse, watch films, or walk through city streets, have revealed patterns of brain activity far richer and more dynamic than those seen in the artificial quiet of the scanner. In these studies, the brain's intricate dance of cognition, emotion, and social interaction emerges vividly, showing that its true genius lies in its ability to operate in the messy, unpredictable, and often exhilarating context of daily life. At the heart of this adaptability is the brain's remarkable power to integrate information. Different regions do not simply fire in isolation, they work in synchrony, weaving together raw sensation, memory, emotion, and intention into a seamless experience. This integration is no accident of evolution, but a feature built into the very architecture of the brain. From the earliest stages of development, even before birth, the foundations for emotional and cognitive life are laid down as connections form, strengthen, and adapt in response to the environment. Children's emotional development, for example, is etched into the neural circuits that will shape how they respond to joy, fear, and frustration for years to come. This ongoing process ensures that the brain remains responsive, never fully finished, always sculpted by experience. Some of the most fascinating examples of this integration occur in the medial temporal lobe, particularly in the hippocampus. This region is not just the seat of memory storing the details of a birthday party or the face of a loved one, but also the engine of imagination. The same neural machinery that allows us to recall a stroll through a neighborhood also lets us imagine wandering through a city we have never visited. The hippocampus supports both real and imagined navigation, orchestrating patterns of neural activity that enable us to move through space and time, whether in the world itself or in the theater of the mind. This ability to blend memory and imagination gives humans extraordinary flexibility, allowing us to plan, dream, and adapt to new situations by simulating possibilities before we act. Yet the organization of the brain is not just about individual regions and their specialized tasks. It is shaped by rhythms and patterns that unfold over time and in relation to others. The temporal dynamics of neural activity. The timing of when different circuits come online, how they synchronize or fall out of step, are essential for making sense of the world. Social interaction, too, is woven into the fabric of brain function. In natural settings, the presence of others changes the way the brain operates. Shared attention, emotional resonance, and the subtle give and take of conversation activate networks that are dormant in solitude. These principles, timing and social context, serve as organizing themes, guiding how the brain processes information and responds to the ever-changing demands of real life. Beneath this flexible surface, advanced imaging techniques have revealed a kind of functional backbone, a set of robust, delay-free connections that form the scaffold for all mental activity. These stable pathways ensure that critical information can be shared rapidly and efficiently, providing a foundation on which more dynamic, context-sensitive connections can be built. Around this backbone lies a vast web of flexible links, connections that wax and wane as circumstances shift, allowing the brain to pivot quickly from focused concentration to broad, creative exploration. This combination of stability and flexibility is perhaps the secret to the brain's resilience, enabling it to maintain core functions while adapting to novelty and change. But the environment in which the brain operates is not always benign. The air we breathe, the food we eat, even the sounds and sights that fill our days can shape brain function for better or worse. Exposure to environmental toxins, whether heavy metals, pesticides, or the byproducts of modern industry, can trigger inflammatory responses that ripple through the brain's delicate networks. The consequences can be far-reaching, from neurodegenerative diseases to neurodevelopmental and psychiatric disorders. These toxins disrupt the brain at the cellular level, interfering with energy production in mitochondria, causing oxidative stress and, in some cases, leading to the death of neurons. The very architecture that enables flexibility and integration can become a vulnerability when challenged by persistent environmental threats. The idea that the health of our surroundings is intimately linked to the health of our brains is gaining increasing recognition. Emerging research in neuroscience underscores the reciprocal relationship between the environment and brain function. For example, changes in climate or exposure to pollutants do not just affect the body, they leave their mark on the brain, influencing how we think, feel, and act. Conversely, the brain's extraordinary sensitivity to environmental cues means that positive changes, such as exposure to natural environments, can have a healing effect. Time spent in green spaces, forests, or even urban parks has been shown to calm the mind, improve attention, and enhance functional connectivity. During these moments in nature, the brain's sensory processing shifts, often leading to quieter, more harmonious patterns of activity. Of course, not all environmental influences are visible. Early life adversity and chronic exposure to stress can leave deep and lasting marks, altering the very structure and function of the brain. These changes can include thinning of the cortex and acceleration of the brain's aging processes at the molecular level. Yet the same neuroplasticity that allows for adaptation to new challenges also offers hope for repair. Physical activity, for instance, has been shown to restore some of the brain's markers that are affected by stress. The brain's architecture, while shaped in part by adversity, remains open to positive change across the lifespan. Scientists now use the term brain pollution to describe the constellation of factors that can erode cognitive function and undermine brain health. Environmental toxins are only one piece of this puzzle. Poor diet, chronic stress, lack of sleep, and sedentary lifestyles can all damage brain cells and disrupt the networks that support memory, attention, and emotion. The cumulative effects of these influences highlight the brain's vulnerability, but also its extraordinary capacity for recovery. By addressing these sources of brain pollution, through cleaner environments, better nutrition, restful sleep, and regular movement, individuals and societies can support the brain's natural resilience. What emerges from all of this is a portrait of the brain as a dynamic, living architecture, constantly integrating new information, adapting to shifting environments, and responding to both internal and external challenges. Its structure is not fixed, but continually molded by experience, shaped by the people we meet, the places we inhabit, and the choices we make. The functional backbone of the brain provides stability, but it is the web of flexible, ever-changing connections that allows for creativity, learning, and growth. In the end, the architecture of the human brain is less like that of a cathedral finished and unchanging, and more like that of a bustling city. Pathways are built and rebuilt, neighborhoods flourish and fade, and the entire system responds with energy and ingenuity to the forces that shape it. This dynamism is not a flaw, but the essence of what it means to be human. The brain's greatest achievement is not simply to process information or control movement, but to integrate, adapt, and thrive amid the complexity of life. And, as our understanding deepens through science, through experience, and through the ongoing dance between brain and world, we come closer to appreciating the full wonder of the architecture within us.